Hello friends, this is Dr. Ravi Sani. Our today's discussion is about various errors or types of errors, mainly type 1 and type 2 errors. Type 1 is called alpha error and type 2 is called beta error. So before we jump to types of error, first we should know where it is used and why it is important. So basically these errors are important in case of hypothesis testing. So first we should know about the hypothesis testing. Hypothesis testing is an important activity of empirical research and evidence-based medicine. A well-worked up hypothesis is half the answer to research question. For this, both knowledge of the subject derived from extensive review of the literature and working knowledge of basic statistical concepts are desirable. So, hypothesis means hypothesis is a very important activity in case of empirical research and evidence-based medicine. So a well worked up hypothesis is half the answer to the research question, because research question is the whole thing around which our thesis goes. So uh, a, a very well precised hypothesis is very much important for that. Now a good hypothesis must be based on good research questions. So the research question should be simple, specific, and stated in advance. So whole the clarity of hypothesis is completely based on the research question. So the research question should be simple, specific, and advanced. So then the hypothesis would also be simple, specific, and would be stated in advance. So various types of hypothesis. For the purpose of testing statistical significance, hypotheses are classified by the way they describe the expected differences between the study groups. Null and alternative hypothesis. The null hypothesis states that there is no association between the predictor and outcome variables in the population. There is no difference between tranquilizer habits of patients with attempted suicide and those of age and sex matched control patients hospitalized for other diagnoses. The null hypothesis is the formal basis for testing statistical significance. By starting with the proposition that there is no association, statistical test can estimate the probability that an observed association could be due to chance. So in case of null hypothesis, we assume that there is no uh, relation between the uh, desirable variables. So if there is any association by chance, which would be reflected in case of uh, the alternative hypothesis. So the proposition that there is an association that patients with attempted suicides will report different tranquilizer habits from those of the controls is called the alternative hypothesis. The hypothesis which is just opposite to the null hypothesis is the alternative hypothesis, which is our research question. The alternative hypothesis cannot be tested directly. It is accepted by exclusion if the test of statistical significance rejects the null hypothesis. Now the type 1 and type 2 error. Type 1 error is also called alpha error and type 2 error is also called beta error. Just like a judge's conclusion, an investigator's conclusion may be wrong. So, sometimes by chance alone, a sample is not representative of the population. Thus, the results in the sample do not reflect reality in the population, and the random error leads to erroneous inference. Type 1 error, which is false positive, occurs if investigator rejects a null hypothesis that is actually true in the population. So, in case of type 1 error, the null hypothesis is rejected in reality, or in actuality it is true in population. Just opposite happens in type 2 error, which is also called false negative. If investigator fails to reject a null hypothesis that is actually false in the population. Although type 1 and type 2 errors can never be avoided entirely, the investigator can reduce their likelihood by increasing the sample size. The larger the sample size, the lesser is the likelihood that it would differ substantially from the population. So both the errors cannot be avoided entirely, but they can be reduced by increasing the sample size. If sample size would be large, then these both errors could be reduced. False positive and false negative results can also occur because of bias like observer, instrument recall, etc. Error due to bias, however, are not referred to as type 1 and type 2 errors. Such errors are troublesome since they may be difficult to detect and cannot usually be quantified.